Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled, The Next Generation of Pathway Analysis for Integrated Omics Data. Omics techniques have played an ever-increasing and important role in gaining a better understanding of various human disease mechanisms. But these same techniques also produce huge amounts of complex data, and researchers are often faced with trying to interpret and make sense of it all. Today's webinar has been designed to help scientists meet this challenge. I'm John Sterling, editor-in-chief of GEN, and I'm going to serve as the moderator. But first, I'd like to introduce our expert panelists who will demonstrate how an approach that utilizes systems biology and pathway analysis can allow researchers to successfully integrate omics data. Chris McKenna, General Manager of Discovery Science at Thomson-Reuters, will set the background for our webinar. He's going to provide an overview of the pharma industry's current R&D challenges and opportunities. Dr. Mark Hughes, Senior Product Manager for Systems Biology and Analytics at Thomson-Reuters, will discuss how the company's METACORE integrated software suite can generate critical biological insights from big data. And Dr. Matthew Wampel is a solution scientist at Thomson-Reuters. Matt will continue the discussion on how pathway analysis can serve as an essential biological discovery research tool. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console, and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. So, if everyone's ready, let us begin. Chris McKenna will get us going. Chris?
Thanks, John, for the introduction. First, I want to start with a little background at Thomson-Reuters. Some on the phone may not know that Thomson-Reuters has been in the business of scientific research, supporting researchers for over 50 years, and in the pharmaceutical industry, supporting pharmaceutical R&D for over 30 years. And one of the areas that we're actively involved in conducting dry lab research within Thomson-Reuters is looking at pharmaceutical R&D productivity and effectiveness. We have on the order of 6 or 7 projects that are run every year, one together with Deloitte, where we look at the key drivers of R&D productivity and how those drivers manifest themselves in terms of financial return on investment in drug discovery and development. One of the outcomes of that research that's fairly well understood is that pharmaceutical R&D productivity continues to drop. However, in the last 5 years, something really dramatic has happened. The return on investment in pharmaceutical R&D has dropped from the mid-double digits, the mid-teens, to just below 5%. And that's had an enormous impact on the financial health of the pharmaceutical industry. So what we do in these programs is we look at the various drivers to gain some insight in terms of different strategies that can be implemented to provide solutions to the challenges that the industry is facing. So some of those insights, some of the strategies that we see being deployed that I'm showing here is that the pharmaceutical industry, even though spending is increasing, most of that spending is occurring in emerging markets. The industry is attempting to launch assets that it has already into emerging markets by conducting studies and gaining market access into these emerging markets. The industry is also being more rigorous about its clinical development and attempting to optimize clinical development in terms of its site selection, optimizing patient recruitment, and reducing protocol development or amendments to protocols, which can extend cycle times and add cost to the development process. On the portfolio side, the in-licensing activity is vibrant, and the industry continues to look for external innovation in academic and government settings that it can in-license and take advantage of new breakthrough therapies and biologics that it can add to its pipeline and continue to develop and launch into the market to treat patients. And lastly, next generation sequencing and big data techniques are being brought into the industry, being implemented in ways to better stratify patients, to translate new findings from early research into humans, and to develop personalized treatments. And so all of these different strategies are being implemented, but it provides insights to ways for Thomson-Reuters and for the industry and others to really attack the problem. Where are the problems in understanding disease, understanding how to bring new therapies to market? And for us, as a supporter of pharmaceutical R&D, it helps us to focus our products and services on those areas that can contribute to the industry. One of the common denominators that exists across these different strategies is improved understanding of smaller cohorts of patients and the disease that they have. And that extends, that's not just happening in research, it extends all across this entire value chain, from research to understanding how animal models translate to humans, to understanding how to the best ways to recruit patients, which sites to select, how to optimize those, how to gain insight into health economic outcomes research, and the commercialization activities that are supported to bring the drug to market. If you take a look at R&D cycle times, it's a real indicator of one of the improvements the industry's made in the last 5 years. And in that 5-year period, by adopting some of these strategies, the large pharma companies have reduced R&D cycle times from 15 years in 2010 to just under 9 years today. So they've dropped 6 years off of the R&D process. Also, during that time, the number of personalized therapies that have gained approval have tripled. So drugs like Herceptin from Genentech Roche, it's a clear example of these strategies at play and the success that they can have. And in that case, a drug that's targeting a specific patient population, HER2-positive breast cancer, and today that drug is achieving close to $7 billion in sales. So these successes are happening, and patient stratification is one of the key areas that we're really focused on as a provider to the industry to support what we see as a key influencer of success in drug discovery and development and bringing new innovative therapies to patients. Now, whether you're operating in a pharmaceutical company, whether you're working in a biotech, or you're a representative of an academic or government researcher, or if you're working in a nonprofit disease foundation, one of the common themes that we hear from our customers is they want Thomson-Reuters to provide flexible ways of working. And in today's webinar, we'll be discussing some of the approaches that we're taking to omics data analysis and patient stratification applications using METACORE. And you'll hear about METACORE and how it's using the various content that we provide to support that application, and also a new workflow in METACORE called the Key Pathways application. Before we get into those talks, though, I want to mention a couple of other approaches that we take, because we believe in this principle of providing openness and flexible solutions. And our vision is that analytics and content should not be locked into a single product. They should exist in more modular forms. And as the academic world increasingly publishes new systems biology methods and omics analytics techniques, keeping up with that innovation is difficult for not only the industry, but providers like Thomson-Reuters when those new innovations are baked into a product. So we've really looked at this and asked ourselves, why not embrace that growth with different approaches, more innovative approaches? And so last year we started a pre-competitive consortium called CBDD, which stands for Computational Biology Methods for Drug Discovery. We have a group of leading pharmaceutical companies that are participating in that consortium, which aims to benchmark and implement, vet, and optimize those analytics. And one of the outputs of that for us is being able to take those innovations and those new analytics and apply them in more flexible ways. You'll be hearing more about that in Dr. Hughes' talk on key pathways application. Our vision also includes the long list of application programming interfaces that we produce for access to our content, providing flexible content that can be easily integrated with other content from other vendors or other public sources or customers' proprietary data. It's an important part of our strategy and an important part of that flexible access to data that we want to support. That's why we embrace initiatives like Transmart, where we provide an end-to-end service in that environment, where we bring our curation and ontology management, our APIs and analytics into that environment to support Transmart implementation. And we're grateful to the community in Transmart that we collaborate with that provide those services. Our vision also includes workflows for processing next-generation sequencing and providing clinical interpretation of whole genome sequencing or specific gene panels to the physicians that are treating patients. And we'll be launching Clinical Genomics Toolkit next year as part of that strategy. So I hope today's webinar demonstrates a perspective into the science at Thomson-Reuters and the ways that we're focusing to provide more flexible access and research methods to support you in your patient stratification and your omics analysis methods. Thank you, John. Back to you.
Chris, thank you very much. As a wonderfully timely and concise overview on the current pharma R&D environment, your reference, especially about the need for improved patient stratification, was very well taken. And that's going to be one of the key challenges, as well as an opportunity for pharma as we move into the future. So thank you for pointing that out. If you are just now joining our webinar, welcome. As I mentioned earlier, we'll be conducting a Q&A segment following the panelists' presentations. Please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console and then hit Submit. Our second panelist, Dr. Mark Hughes, will now begin his presentation. Mark?
Thank you, John, and thank you, Chris, for a good overview of Thomson-Reuters. Okay, so I'm going to sort of pick up on some of the things that Chris mentioned in his overview and focus a bit more on some of the aspects of content, the tools with METACORE, and some of the future sort of developments and workflows that we're building at the moment. So to start this, we'll talk about some of the challenges that are faced in discovery science with generating biological insights from the big data that is now being generated so readily. So as Chris mentioned, the pharmaceutical and indeed academic and large disease research institutions are facing a number of challenges in how to analyze and look at this data. And these are around identifying targets and pathways to address unmet medical needs, identifying potential drugs and target combinations that should focus on their sort of therapeutic projects. Another big main driver through all of these sort of challenges is which patient population have the best treatment response versus non-response for a given target or candidate. And again, with the biomarker discovery to distinguish patient populations and support this therapy development. All this allowing, and all these sort of challenges, half the difficulty is allowing sort of the discovery biologists to access these analytics and information through an easy-to-use interface. Lots of these sort of algorithms and methodologies are sort of developed in academia and are not really very accessible to the sort of discovery bench biologists. So within Thomson-Reuters, we've got a number of products that are built to address some of these problems. So we have METACORE, our main sort of systems biology pathway analysis suite, which we'll be talking about in more detail. And then behind that, we have things like METABASE, which is our database of content, MiniBase, which is a sort of a smaller version, more focus of that, so you can get the information around particular therapy areas, and other products like MetaDrug and new disease research advisors and the APIs coming along. So as well as the products, as Chris mentioned in his part, that we also, focus on the content, but also on the professional services. So we have a big service division that develop bioinformatic algorithms, do disease mechanism reconstructions, target and biomarker discovery, patient stratification. And as Chris mentioned earlier on, the computational biology for drug discovery program. This service department is very key within the organization. It gives us real insights into new algorithms, new applications, and how we can actually implement these within a particular workflow or drug discovery pipeline. So focusing on METACORE. So METACORE is based around the sort of annotations underlying the system. And Thomson-Reuters being a content company, this is something, this high-quality content manually curated is very key to the systems. And we'll talk a bit more about driving your analysis through data and through knowledge. So within the back end of METACORE, within the system, it is manually curated from journals, from full-text articles, including figures and images within there. It is curated by PhDs and MDs who are specialists within their particular subject area. We capture a high level of annotation and quality. We focus around those small experiments where we have validation rather than the large throughput experiments that can have a lot of noise around the data, as we know with the issues with big data at the moment. So each of our annotations, they have 3 months training on just how to sort of read a paper, annotate and extract the information from that. And then we have 3 levels of supervision and quality control over the top of that. So we're really striving to have the best quality content within there. And this is important as you move forward with your analysis. So all this information we have stored in Oracle tables. It's all connected by controlled ontologies and controlled vocabulary. And again, that allows us very much to connect the content between the different parts of Thomson-Reuters. And again, as mentioned earlier, it allows you to add those extra insights on top of your data as you go through your analysis. So again, we have multiple ways of accessing that, and here we're going to focus really on METACORE. And again, this is the UI access to all of this knowledge and content, and providing that analytical layer to allow the biologist to extract knowledge from their data. So focusing a little bit more on the content and how this is constructed so you can understand what is going into the back end of these systems to run it. We extract from the publication these molecular interactions. So here we can see EGFR with EGFR receptor, binding interaction in green meaning it's activation, and we have it directional as well. And again, this is important when we come to some of these advanced analytics, having this layer of information above this interaction with the directionality, the effect of it, and potentially the mechanism as well. From these interactions, we can build large molecular networks. We have over 1.5 million molecular interactions within the networks within these databases, and these often refer to these sort of hairballs that we know. But part of our process of annotating the content in this system is we build a lot of pathways. So these represent, we have over 3,000 pathways at still. These represent linear pathways which start off with maybe a signal going down to an effect. And these are very well documented, very well understood mechanisms and pathways. So using all that knowledge from the literature to really understand the mechanisms. So as well as around these pathways around particular mechanisms, we have these around particular diseases as well, giving a real insight into the mechanisms and how your data overlays on a particular disease. So the approach we've had with METABASE over the last 14 years curating this, we believe we have the best content to allow you to apply these analytics over. And it's not just that, there's a number of independent studies and papers that have been published around this, highlighting the breadth and the depth of the content within METABASE that all of our analytics are based off. So in this new era of NGS data, we produce lots of genomic-wide information around sort of epigenetics, about the DNA variants within there. And all of this can be used in the workflows for drug target identification, disease mechanism reconstruction, biomarker discovery, drug repositioning, and drug combination. So it's really opened up major opportunities, but as we say, major challenges with analyzing this data. So to talk about sort of the Thomson-Reuters approach, we very much believe using prior biological knowledge for disease understanding and cure, and analyzing your data that you're generating through a data-driven approach, looking at various models. Around that, we talk about some of the algorithms that are being coded up within the CBDD, but also using that knowledge-based approach, using those pathways, using those molecular interactions to really understand the big data. So in some way, using those small data, those interaction, to understand what's going on in the big data of your NGS analysis data. So this is not something we're just doing internally. We are doing this in collaboration with pharma companies and biotech companies. Last year, 2013, we announced a partnership with NewMedi, where we're applying a lot of these sort of algorithms and big data technologies to identify drugs for repositioning. So again, it's not something we're just doing in isolation. We're doing this in collaboration with some large partners. So to talk a little bit about one of the projects, and just a couple of slides to overview this, because this is the knowledge we're gaining from this is really driving some of the innovation that's going to be moving into the products as we move forward. So the Computational Biology Tools for Drug Discovery program, this is a program with, I'd say, a pre-competitive program with large pharma companies to really look at all the different sort of algorithms that are available out there within the domain, to evaluate them in their utility, their biological, their biological utility, and understand how they operate. So we are coding up a number of different algorithms, and one of the things that's sort of really come out of some of this program, that different algorithms are useful for different types of workflow and answering different questions. So we understand there's a group of algorithms that are very good at looking for target prediction and repositioning. And again, there are other algorithms that would be more based around identifying sort of biomarker discovery workflows. So all of this knowledge that we've gained within these sort of pre-competitive collaborations that we've done, we're starting to now use that to feed into the products, so we can really build some very, very powerful workflows to answer some very specific challenges within the industry. So again, just highlighting, this is something we are also trying to put, also putting into practice. The Services Department, METABASE, published in 2013 a paper which we looked at drug repositioning, drug target prediction and repositioning for 30 diseases. So this was really a data and a knowledge-driven approach. We accessed microarray data from the public data sources, using our own interaction information within METABASE, and using a variety of network analysis algorithms, and also using a consensus classifier that was powered by our integrity database, which holds information about biomarkers specific to diseases. It allowed us to predict drug targets for each of the 30 diseases. This method was shown to predict the drug targets for these diseases with a high performance, with up to over 90% of known drug targets for some diseases predicted by this method. So as well as using this for drug target, for identifying those known drug targets, part of this work also looked at those other potential targets that were identified within this analytical workflow. And when we look at this graph on the right, these sort of darker blue on the left-hand side, these are the known drug targets. The red ones in the middle, these represent unknown potential drug targets at the moment. So these are not in any terms sort of being investigated. But the bars on the right-hand side, when we looked into these drug targets, these are all either in early stage, late stage clinical, or in discovery. So as well as identifying those known drug targets, we also managed to predict a high percentage of the drug targets in the development at the moment, and also with the potential of a whole number of other targets that could be used for investigation as potential targets for that particular disease. So again, moving back in, we've got the problem with the NGS data and the challenges that presents within within that. So now moving on to the new application that we are going to be releasing later this month. And this is our METACORE Key Pathway Advisor tool. So this is a companion application sitting alongside METACORE. It's a sort of proof of principle of the new advanced workflows and easier interface to the data to really allow sort of the discovery scientists to extend their analysis into some very advanced analytics. So there are a number of challenges we try to address with the Key Pathway Advisor tool. The idea is it to be easy to use workflows to enhance and expedite research for the discovery biologist. It needs to have an advanced pathway analysis capability, standard sort of gene set enrichment and pathway analysis. Generally didn't give a succinct enough answer within there because of noise within systems. Another key driver with this tool is to combine key hub identification with pathway enrichment. So this is one of the key areas of this tool, and we'll talk a little bit more about the algorithms as we move through. But here we have implemented the causal reasoning algorithm and an interactome algorithm with the potential to extend these out with further algorithms from the CBDD program as we build out new workflows. The idea of this building these enrichments around these key driver hubs within your data is it's going to generate greater biological significance in the results. So it's going to focus those pathways and mechanisms on where you've got genes that are highly connected and seem to be controlling downstream changes. And really, with this system being connected to METACORE, it's going to give you the ability to dive deeper into their data to gain more detailed insights. So this is the new easy-to-use workflow. It starts with an input list. We do standard enrichment with that. We also then take that input list and we move right along this workflow. And here we apply the causal reasoning argument. So this is, as well as looking for the actual nodes being in there, it's also looking at the directionality and the effect of the interaction between them. So it's a real data-driven method. And the output of this is the key hub. So it's identifies a list of those objects within your data that appear to be controlling the sort of downstream expression of that. And then by doing various enrichments from those and looking for synergy across these, we come out with a key list of pathways and any of the other ontologies as well within that are available within METACORE. Come out with a much more succinct list of those that hopefully highlight the true biological mechanisms within your data. So it's really enabled through a new, much simplified interface. So we have the ability to just drag and drop our input files onto it. We have a few settings that we need to select, where we can select the algorithm and some p-values, and then we run the analysis, and it generates a PDF report for you to read at the end, which is linked back to METACORE. Once you've loaded your file on, I say it's a very simplified workflow. It leads you through it. You can choose here the various different ontologies that you would like. And down at the bottom half in this advanced settings of the panel, here we can choose the type of algorithm and the p-value that we want to run here. So within this current implementation, we've implemented two algorithms. We've implemented the causal reasoning, forward and reverse causal reasoning. So this obviously takes into account your expression data within the interaction to see if it confirms with the expected expression change with what's known about the interaction between two data objects. The other algorithm we have in here at the moment is an interactome, which looks at over-connectivity of any object within your list. And this can be powered when you don't actually have any expression values. So we default to the causal reasoning when possible, but we use interactome where we don't have necessary directionality on our data. So at the end of this, you'll get an email giving you a link to your report in the account. And here we have just a few screenshots from the report. So we have all the key pathways. We give you the top 100 within here at the moment. These are hypertext linked back to METACORE. And then we have various p-values associated with them for their significance against the input list, against the key hubs, and the union of those lists. So some various sorting methods within there, depending on which which of those lists is more important to you. As mentioned, this is linked back, so we're going to click through the document. You can be back in your METACORE account, and here we can view any of those maps, and as long as the data is loaded into METACORE as well, we can view on here the thermometers, which will show us where we have the data overlapped. So the red thermometer being overexpression and a blue thermometer being downregulation. So as well as the pathways, the other key part of this report for me is the identification of these key hubs. So this is the output from the causal reasoning algorithm. And here we have all the network objects. These are ranked by their significance and their p-value. Within here, it tells you a little bit of information about what the molecular function is, the activity, positive for upregulated, negative for downregulated, the overall prediction. So this top one, 34 of the objects downstream from this out of 41 were following the expected expression pattern from the known interactions and the p-value. And then we have the p-value and the number of steps down from there. So these are the real potential drivers within the data of your experiment. So from here, we can very quickly also go back, take that sort of information back into METACORE, and we can start building the downstream network from those hubs. So in this particular image here, on the very left-hand side, we can see that top driver hub. And downstream, we can see it's connected to a number of objects, and each of these objects has a red halo around it, saying that it's within our starting list. And they also have some circles on there for red for upregulated and blue for downregulated. And when you go in and examine this, you can really identify those 34 objects that are following the expected expression pattern if they're being regulated by this particular object on the left of the map. So it really is allowing the biologists to build these very focused networks that are showing gene expression patterns that match what we're expecting or match what we're expecting from the disruption of that particular mechanism. So where can a biologist take these? And again, Matt's going to sort of go on a little bit more and extract how we put this into answering a biological question. But really, once we've got to this point with the key pathway advisor, we've identified those key driver hubs within the data. So these are probably the most important genes within the data set because these are, let's say, driving the changes downstream. But from those pathways and networks, we can start looking for possible causative pathways. We can start identifying possible druggable targets around those pathways using the various other content overlays we have within Thomson-Reuters. And again, also be able to identify possible biomarkers from those pathways. So some of the overlays we have is things like tissue specificity, whether it can be found in a fluid. So we can identify biomarkers that could potentially be screened for. And also these pathways that are coming up could be very important in moving into that sort of patient stratification. So the key pathway advisor is really the first workflow we're releasing along this path of producing more of these workflows to answer very, very specific challenges within discovery science. One of the next sort of advances from this will be the implementation of a similar type of workflow, but over multiple omics data types. So just to sort of wrap up, let's say thank you, and I'm going to hand back to John, but you can also find out more information from the Life Science Research website at Thomson-Reuters. Thank you very much.
Hey, thank you, Mark, for describing how a number of Thomson-Reuters software tools and approaches can really provide significant biological pathway information and really assist in many areas of the drug discovery process. There was lots of practical advice there, so thank you again. Before we proceed, let me remind everyone of our Q&A segment that comes right after the panelists made their presentations. Please type your questions into the Ask a Question box on the lower left of your console and then hit Submit. Our third panelist, Dr. Matt Wampel, is ready to get going about a case study involving sophisticated genomic analysis tools. Matt, it's all yours.
Thank you, John. For my portion of today's talk, I'm going to provide a brief discussion on using multi-omics approaches to gaining biological understanding in colorectal cancer relapse. Next-generation sequencing is a great boon for gaining new knowledge of disease and drug discovery through patient stratification and understanding of causative mutation. Two big challenges facing researchers who want to take advantage of this new technology are size and complexity of the data. For a single sample, the output from next-generation sequencing can generate over 10 million data points in a file of up to 2 gigabytes in size. So large-scale experiments can include hundreds of samples, leading to challenges of storing and analyzing this big data. Earlier in the year, we released a new tool for METACORE called the Genomic Analysis Tool to help alleviate these challenges and provide researchers with the tools to make the next big discovery using next-generation sequencing. METACORE is a web-based portal that allows you to upload and analyze multiple types of omics data. The Genomic Analysis Tools expands these capabilities by allowing users to upload gene variant files from next-generation sequencing, keeping these files available at any computer at any time via a web browser. It also empowers the researchers with statistical tools and curated knowledge to filter those millions of variants down to those causative variants. Once filtered, they can then use that list of variants in pathway analysis and network building alongside their other omics data. Using the genomic analysis tools, you can annotate the variants, run functional predictions, and use pathway analysis to understand the impact these variants could have in biology. Annotations from manually curated content by our team of PhDs and MDs gives you a rich and highly indexed set of knowledge to identify variants with known phenotypic impact. When discovering a new variant, using functional predictions can really help to identify which variants may be damaging to the overall function of a particular protein. Finally, using pathway analysis, one can identify what signaling cascades and overarching processes may be significantly disrupted by these variants. METACORE allows you to not only perform pathway analysis on the variant, but on other omics data sets simultaneously. This is important since diseases are rarely caused by a single alteration in a normal process. By looking at the biological changes on multiple levels, one can get a better sense of what changes might be key drivers of disease and develop targeted treatments for it. In this case study being presented here, gene variant and transcriptomic data sets will be analyzed to look for molecular features that will help determine relapse in colorectal cancer. The gene variant data comes from a publication by Medhaven et al., who are using the data set as part of a multi-omics profiling of patients to identify immune determinants that are associated with relapse in colorectal cancer. The transcriptomic data set comes from the GEA repository and was incorporated into our microarray repository in METACORE. Just to let you know, the microarray repository is a collection of publicly available data sets that we've gone through and done quality control checks as well as calculated fold change, and it's available for users of METACORE. Now, the first step to analyzing the gene variant data is to perform an analysis to determine significant variants in the group with colorectal cancer relapse versus those who are relapse-free. To do this, I'll use the cohort analysis option within the genomic analysis tool to find those significant variants using a Cochrane-Armitage trend test to compare the samples between the two groups. So we also have options to perform other types of comparisons for looking at diseases between normal tissue and diseased tissue in the same patient, as well as performing a pedigree analysis to find inherited genes passed from parent to child. The next step in the process is to annotate and filter those variants to produce a list of potentially causative mutations. I'll first want to apply a filter based on the p-values calculated from the cohort analysis to identify any significant variant. This is a good starting point to begin looking at those causative variants in the data set, and then apply other filters or moving on to pathway analysis. Now, some of the other filters that are available within the genomic analysis tools include other types of chromosome location, as well as filtering based on the content from METACORE on what genes or proteins associated with particular subjects, such as diseases, where they might be expressed in tissue types, or their roles in various biological processes. A new content area for METACORE that's provided through the genomic analysis tools gives you information about gene variants associated with different types of diseases and drug treatment. And of course, you can also filter by the type of mutation, prediction of the impact of the as well as if the variant is present in any conserved regions, or just how novel the variant is in various populations. Talking a little bit more about our new content gene variants, we've gone through and curated a number of subjects around these variants. You can find relationships between these variants in your data set and publish records on how these variants are associated with diseases or response to various treatments. These disease records break down the association into specific areas such as variants being used in profiling, staging, or diagnosis of a disease, as well as looking at response to treatment, which can be further broken into two areas, looking at the efficacy or toxicity of treatment. This can be very useful for a researcher or others who might be looking to pick a treatment for a type of group of disease or predicting toxic events that might occur due to a treatment. Now, applying an additional filter to my p-value filter, I can take a look at what genes in that list are associated with treatment efficacy. From that filter, I identified 5 variants in particular. But one of those variants is related to the protein PCSK9. We take a closer look at this variant and at the response record for it, we uncover that this variant has been associated with treatments using statin to treat hypercholesterolemia and myocardial infarction. Digging even further into the record, you can find that the statin treatments in these patients with the variant have been ineffective. So since this variant is identified as being significant within our patients who have relapsing colorectal cancer, we could predict that, in general, these patients may find that statin treatments are generally ineffective for them. If you go back, we can apply a different filter, but this time looking at the treatment for toxic identifiers within our variant. We find that there is a variant related to the major histocompatibility complex class 2 gene. And looking at this treatment response page, we find that this variant has been associated with the drug lepatimid and is connected with induced liver injury due to the treatment in patients with this variant. You can also take a look at other diseases associated with this disease. We find quite a bit of striking information here. For this particular variant, it has been associated with quite a number of other diseases, many of which are associated with inflammatory bowel disease and other types of gastrointestinal diseases such as celiac. Now, taking our filtered variant and then incorporating it with our transcriptomic data, we can paint a bigger picture of some of the biological processes being disrupted in patients with relapsing colorectal cancer. Performing pathway analysis on these 2 data sets identifies this signaling pathway as being significantly disrupted. This particular map looks at signaling due to IL-1 and how the signaling process progresses down through the cascade and leads to transcription factors that are related to the expression of pro-inflammatory cytokines. Looking over the map, you might notice that a number of indicators related to our data set are around the transcription factor. A closer look at these indicators, you'll notice that the zebra stripes here are indicators for our variant data, and they are both attached to the IKK beta protein. And when we look downstream from these proteins with variants associated with them, we'll notice that IRF1 as well as INOS have blue thermometers associated, and these are related to our expression data, and the blue indicates that they are being decreased in expression. IRF1 and INOS are both transcriptional targets of NF-kappa B and STAT1. So it could be hypothesized that the variance in IKK beta and the decrease in expression in STAT1 may be related to the decrease in expression of IRF1 and INOS. Now looking further into some of the disease records associated with IRF1, we can find some association with colorectal cancer relapse patients. In particular, this paper here, which was published, states that high expression of genes, including IRF1, is associated with significant improvement of disease-free survival in patients with colorectal cancer. So the fact that we found that these two, IKK beta, as well as potentially STAT1, may be inducing a decrease in IRF1, may be indicators of relapse in colorectal patients. Let's summarize some of the things we've learned from analyzing patients' gene variants and expression data. The results suggest that mutations in PCSK9 could lead to statin treatments in colorectal patients and may be ineffective after relapse. Damaging mutations in MHC class 2, common in other gastrointestinal disorders, such as celiac disease, Reckitt's disease, and ulcerative colitis, as well as potentially damaging mutations in IKK cat complex, which could inactivate promotion of NF-κB target genes in response to IL-1 signaling. And this can lead to decreased expression of the pro-inflammatory cytokines IL-6 and IL-8. So through this presentation, we looked over how you can start with transcriptomic and gene variant data and perform statistical and pathway analysis to gain a better understanding of the biological mechanisms, biomarkers, potential drug targets, or concerns in relapsing colorectal cancer. With that, I just want to say thank you for joining us and part of the presentation. Now, back to you, John.
Well, thank you, Matt, and thanks again to Chris and Mark for your presentations. The focus of today's webinar was to take a close look at next-generation pathway analysis tools for integrating genomics data. I think each of you did a superb job in helping us meet that goal, so much appreciated. Shortly, we're going to begin our Q&A segment, so feel free to send in a question for either of our panelists. Type your question into the Ask a Question box on your computer console, and then hit Submit. The panel will try to answer as many questions as possible. We actually do have some very interesting questions, but before we start the Q&A, I'd like to ask the audience to disable your pop-up blockers. A short survey on this webinar will be appearing right now. We would very much appreciate your feedback on the webinar so we can continue to bring timely and informative webinars in the future. Let's take the first question from our audience.
Chris, question for you. Are your curators full-time? I really think the investment here is worth it.
Yes, thanks, John, for the question. Yes, so the answer to that is yes. We have curators that are full-time. They're distributed throughout the globe. And in fact, the bulk of our life sciences staff are these scientific curators and scientific analysts, and they're curating from a number of different primary sources, so that it's from the patent authorities, the scientific literature. We have these curators and scientists attending scientific conferences and collecting information off of the floor, reading posters. So all that's done by our employees and highly trained curators and scientists that then insert all that information into a Six Sigma quality-controlled process that then pumps all that information into our products so that it's available through any of the respective products to our customers.
Okay, Chris, another one. Are there any services where one can submit proteomic data of cancer cells treated with one of two different compounds, and you assist with determining the mechanism of the two compounds and the differences between the two?
Oh, yes, excellent question. So we have, within our professional services, a systems biology team that does that type of work, and so we provide services that we call mechanism reconstruction. And we conduct a number of these types of projects throughout the year where you submit the data to us, and then we perform that mechanism reconstruction and provide back the likely mechanisms that the single drug or the multiple drugs are contributing to.
Thank you very much. And Mark, within Key Pathway Advisor, one can bring together multiple types of data like transcriptomics, proteomics, metabolomics into a single analysis. Guess that's a question?
Hi, John. Thank you very much for that question. Yeah. So currently with the Key Pathway Advisor, it's sort of working around the sort of the one list at the moment. So that could either be sort of your transcriptomics or your proteomics or your metabolomic data. Obviously, within the up and coming versions, we're looking at releasing a more integrated data analysis where you'll be able to combine those and use various different data types to drive the sort of causal arguments within there. Obviously, already currently within the sort of full METACORE project, we are able to combine sort of transcriptomics, genomics data, metabolomics, and proteomics data already.
Thank you, Mark. And another question. Would you say more about how causal reasoning is being done? How do you distinguish between correlation and causation among experimental variables and observables?
So yeah, another good question. So the causal reasoning we sort of implemented in this particular workflow comes from the original sort of Pollard paper of a forward and reverse causal reasoning. Obviously, this is a later iteration of that. So one of the sort of the key features of the way the whole systems work is integrating that multiple data types, so you can actually look to see if there is a real true causative effect downstream from that. And also, again, as we sort of move forward into the future, we sort of mentioned the CBDD in there. We are investigating a whole raft of other algorithms that could also provide extra insights within those particular issues.
Okay, Mark. And please define interactome, mentioned used in parallel to causal reasoning. Does TM have a specific product for the interactome?
Yeah, so the interactome analysis is an interesting one. This is an analysis we've had actually for a long time within the METACORE product. And it looks at sort of over-connectivity within your data set. So it's very good at uncovering objects outside of your initial list that are really highly connected to your data. So this has been available for a long time within the METACORE product. And where we use it, particularly within the KPA, is the thing that because the interactome works on just sort of numbers of interactions, it doesn't necessarily rely on sort of expression values up or down. It allows us to apply this type of analysis where you don't have say, directional information about the data.
Thank you, Mark. And Mark, continuing, can you integrate methylation, miRNA, or other omic data, or is it that just genomic and mRNA?
So currently, as I say, within the product, we can integrate sort of genomic, transcriptomic, proteomic, metabolic data, post-translation modification data from proteins as well. One of the things we are looking to in the future at the moment is more that regulatory and epigenetic information. So we're looking at sort of integrating that more deeply in the coming year.
And Mark, please tell us how you are integrating phosphoproteomics data into METACORE.
So as mentioned, we already sort of have quite a lot of sort of phosphoproteomic data inside of METACORE. And again, this year we've got a program where we're looking at a number of different databases and sources to supplement this, and are quite developed in this investigation at the moment. So again, this is one of the areas where the sort of the network object model that we use works very well within METACORE. It allows us to describe sort of a protein and various sort of relationships to that protein, which could be these modifications, and describe a lot of information around those.
And Mark, question, can a key hub be a chemical?
So yeah, no, that's a very good question. So with all of the sort of algorithms within inside of the Key Pathway Advisor and within METACORE, it works on the sort of network objects in the database, and those network objects can be, a chemical, a gene, or a protein, or any of the other sort of entities that we deal with. So chemicals are treated as equally as a gene and a protein.
Okay, thank you very much. Matt, how do you consider the interactions between the genes? You mentioned you use gene set enrichment, but that assumes each gene to be independent.
That's correct. The interactions is what we pull from the various pieces of literature and have our curators annotate. So we look for the specific interactions between various proteins and other objects for the interactions themselves. On the other hand, gene set enrichment is more looking at a collection of genes and what may process they might be related to. So there's a little bit of a difference between the interactions and an actual gene set enrichment.
Okay, thank you much. Matt, have autoimmune diseases been covered in the database, and how extensive?
So the database, at Core, really looks at the regulatory basic processes going on. But we do something called metaminders, where we go out and we look at more specific disease-focused or area of interest that researchers might be interested in collaborating with us on. So this can cover things such as asthma or cancer, but one of the things we're actually doing right now is looking at immunology, so many things like lupus and other autoimmune diseases, and developing pathway maps and other kind of information around those specific diseases of interest.
Okay, Matt, another question. This one's a little long, so bear with me. How can I upload my own data to METACORE? It is not an experiment. It's actually a literature review for all the interactions of G6PD with other proteins and genes. Is there any special format for that, and how can I later put them into a network?
Oh, that's a really good question. I mean, a lot of times people read these articles and they find something brand new they want to try and look at in METACORE, or they have their own data sets, looking at your own transcription factors and looking at the targets. So what you can do is upload a list of just those genes into METACORE, just a simple list, and then analyze them. Or you can use another function within METACORE called MetaLink, which allows you to build interactions between different proteins. So this is really great for those researchers who are discovering brand new interactions and be able to build networks and interactions specific to the research.
And Matt, a question, how do you combine genomics and transcriptomics genes for pathway analysis?
This is a good question. So what we do is we start with the genomic data. So we have all those variants, and we associate them to the gene in question. And then you can take that gene and look at it on the various pathway maps or networks and kind of get a idea of where the variants will be influencing, perhaps damaging the function of a particular protein, and look if there's any downstream or upstream changes of expression from the transcriptomic data that might also be inhibiting or activating those kind of events. So it gives you a much bigger picture of what's going on within the disease.
Okay, thank you, Matt. And question, what data format does METACORE take?
So we can take quite a few different types of data. So for the gene, proteomics, transcriptomics, we can bring in different types of gene IDs. So this can be your IDs from like an AFI chip or an Illumina chip, and you can import the data using those IDs, but you can also use other database IDs, such as for miRNAs, you can use the mirror-based IDs, accession numbers, or you can use Entrez gene IDs or any other number of different gene IDs to upload your data into METACORE. But you can also bring in data from metabolomics. So you just need information such as the Cas number or a SMILE string to help identify the what metabolomic data it is. So as long as you can bring your data in using one of those IDs, you can analyze it in METACORE.
Thank you, Matt. Mark, question, is Key Pathway Advisor an online program?
Yes, so the Key Pathway Advisor is an online system that's going to be made available to METACORE users and other people within their account.
Thank you very much. And we've got time for one more question. I'm going to send this to Chris. Just bring this up here. I'm reasonably familiar with your systems biology and drug pipeline content, but when did you start offering gene variant content, and how is it different from public sources like ClinVar, Cosmic, DD, SNP, and 1000 Genome?
Okay, thanks for the question. Yeah, so I think the way to think about it is that, first we've been, in the first part of the question, we've been curating gene variant content for several years, and those have been parts of various products that we have. And as the association of gene variant to disease has just exploded in the literature recently, we've decided to really invest in creating a source of gene variant content for both life sciences, drug discovery and development purposes, but also for clinical interpretation purposes. So what we do is we, in fact, ingest all of those sources that you mentioned in the question into our curation process. And so they all come into our process. And then what we do is we deduplicate the records. In some of the sources, you'll have one synonym used to describe a gene or gene variant. In another source, you'll have another synonym used. So part of the value we're providing is just deduplicating and creating one single source. But then what we do is we add other types of curation and data on top of that data, as well as add to the data that we're collecting out of the scientific literature. So these are concepts like strength of association, or the effect term, the directionality of the variant association, the validity, or whether there's supporting or conflicting evidence, the power of association, looking at the p-values from the studies and the study type, population information and biomarker information that exists in the studies. So we really embellish and color, add additional color to that content, and then that's part of what we're providing there, and really integrating and taking advantage of the body of knowledge that exists there.
Well, Chris, thank you for the very detailed response. Unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends, which we highly recommend. Thanks again to the panel for the outstanding presentations, and I want to say thank you to our audience for your attention and your very thoughtful questions about the topics brought up during the webinar. And thank you to Thomson-Reuters, who made this webinar possible. Bye for now.